All right, hello everyone. My name is Allison, and I'm going to be presenting on my manual, which is called Visualizing Mass Spectrometry Proteomics Data in R, and why you should use it. So, some background. First is what is proteomics. Proteomics is the study of the structure and function of proteins, which is produced by an organism. Mass spec is often used to perform large-scale proteomics experiments because of its high accuracy and also the fact that this instrument can collect large volumes of data with very small sample sizes, which can be useful in, if you have only a little bit of cell culture but still want to understand what's happening in the proteome. So mass spec uses mass over charge ratios and measures the mass over charge for ions which fly in the mass spectrometer. The mass over charge values are used to calculate the mass of each peptide, which is measured by the mass spec. And you can use other proprietary software to correspond these masses to peptides. So proteomics experiments are used for multiple different kinds of experiments. One is drug discovery. You can see if your compound has actually bound to a protein by looking at changes in mass. You could also have different drug conditions and see how protein expression changes. You can also verify transcriptomics data. So if you have a particular gene that's, maybe highly expressed and you want to confirm that this is true, you could look at mass spectrometry data and see if this protein is present in the proteome as well. You can also look at protein-protein interactions because mass spectrometry can identify dimers, so proteins which have bound together. So it's a very important field of study for multiple reasons. The main question that this manual will answer is, how can mass spec proteomics data be visualized using MS-Stats package and also ggplot? So the MS-Stats package is used for normalizing and processing your mass spectrometry data. You can also create some plots using this package, but in this manual, we're going to use MS-Stats to normalize and process our data, and then create a data set with our mass spec results. And this data set will then be used in ggplot to make our plots. So this manual will demonstrate how to use MS-Stats to quantify the peptides using the peak area of the unique ion which results from each peptide, and then how to visualize the data using ggplot. And we also do some MS-Stats plots in order to look at the quality of our data. So some more background on the data set. This manual utilizes a data set which was developed by Choi et al. And is also provided by a tutorial on GitHub. This tutorial teaches you how to use MS-Stats, but the difference is in this manual, we are going to use MS-Stats to give us a data set which we can use in ggplot. And then we're going to use ggplot to create plots. So for this data set, samples were ran on mass spec, and these samples come from strains of yeast which were mixed with controlled mixtures of protein. So there were 4 protein mixtures created, and these create our 4 conditions. The conditions were ran in triplicate, and then the raw data for this manual is in the form of a Skyline output CSV file. So this file is from the proprietary software known as Skyline, which assigns peptides to masses. So I chose this data set because it mimics a mass spectrometry proteomics experiment data set where the abundance of each protein in the sample is unknown. But in this case, it's known. So it's good for practice because this data will be a bit more clean, that replicates will be accurate and protein levels will be accurate as well because they're more controlled. So in a normal proteomics experiment, you're trying to determine how the protein content changes in each condition. So we're going to see what that will look like using our plots. A little bit more information on the data set. So this is just a screenshot of some of the code which you will, write when you use this manual. So we're looking at the first 6 lines of the raw data set, the Skyline CSV output file that we have. So we have the protein name, we have the peptide sequence, we have a little bit more information that the mass spec gives us, the precursor charge, the M/Z value. We have the condition which this protein corresponds to. And then we have, most importantly, we have the area. So this is the peak area. This peak area is measured by the mass spec and directly corresponds to the abundance of this protein in your sample. So an overview, we're going to process and normalize our data using MS Stats. We're going to then extract our protein level data. We're going to use ggplot to create a data set visualization plot, which is going to look a little bit like a Manhattan plot. Then we're going to use MS Stats again to create our QC plots and our profile plots to do some QC steps on our data. And then we're going to use ggplot to create a comparison plot for a protein of interest. So the first plot we'll look at is our MS Stats QC plot. When MS Stats normalizes the data, it normalizes the replicate and condition across replicates and conditions, and it normalizes to the mean so that all the replicates and conditions have the same mean. So in this case, you can see that, it looks like our data, it looks good and it is normalized because most of our data, falls within this box plot, and there are a few outliers, but we can look in to see why that might be using our condition comparison. Maybe some proteins fall, far outside of this because of the different conditions, and we'll look into that. But yeah, you just want to see that all the means match up, and we see that that's true here. So our normalization looks good, and we can also see across replicates if our replicates look good and if they correspond to one another. So this is a profile plot which you make in MS Stats in this manual, and you can see that the replicates for this protein and in these conditions look good as well. It's pretty common that a replicate might fail in the mass spec because of the mass spec malfunctioning, or there's some sort of issue. But in this case, it looks good. But this kind of plot would tell you that maybe you need to run the experiment again, or you can't really trust this data because one of the replicates does not look good. So getting into our ggplots, this is the data set visualization. So this is all the proteins. And in this manual, we are going to assign each protein a specific number in order to create this visualization. And so you will also learn how to create this vertical line so that all the plots for each protein, the log intensities for each protein, line up, and you can view each protein a bit more easily. And we also add this average log intensity line, so you can get an idea of your distribution. And then we add annotations. So we add the number, which corresponds to the protein name. So you would have to look up this number in your data set to find the protein name. And we do that because if we had the protein name listed for each of these plots, it would get pretty busy. But you could look in and look for, proteins which you're interested in using this data visualization. And then we have our condition plot. So we looked into this protein, which is CW14. It's a glycoprotein which is present in the cell wall, which is present in yeast. So we have our 4 conditions. We have our 3 replicates in black, and then we teach you how to add this average, which shows you the average for each condition, dot in red. So this allows you to look at how protein abundance changes within each condition for a specific protein of interest. So With this manual, you are able to visualize the normalization of your mass spec data. You can see consistency in your replicates for your conditions for a specific protein. And then you're able to create visualizations, which allow for the identification of specific proteins. In the dataset visualization, you can look at maybe outliers in log intensity or look at proteins which are really dynamic, across your entire data. And you could think about which proteins you might want to look into. And I believe that this, dataset visualization is a really good primary step in your data analysis of your proteomics experiment. It allows you to understand the dynamics of the proteins in your sample and might give you some ideas of what to look into. The condition plot allows for visualization of a specific protein across all conditions, which is really useful and could be a next step. So yeah, I think that the advantage of this manual is that it allows you to use ggplot to visualize your mass spec data. MS Stats is used for the processing and normalization, but ggplot can be easier for most people who use R to use and can make it easier for you to understand the steps which allow you to create these plots. And maybe you can customize your plots because you might have a better understanding of ggplot. So yeah, I think that you should use this manual to analyze your mass spec data because it uses ggplot. So these are just some references, and thank you.